Now we're going to move on to section 4. So we know that we have made ATP, right? Okay, that's great. ATP is made in the matrix, right? But now we got to think about what's going to run it. How are we going to get everything into the right locations within the cell, right? You got to think about, okay, if I have electron carriers that are running this electron transport chain to then run ATP synthase. Well, I can make some NADH in the mitochondria, right, in the citrate cycle, which is really easy. It's in the right place. But I also make NADH in the cytosol. That can be through glycolysis and some other pathways that we'll talk about later. And we know that if we deplete NAD+, we're going to halt glycolysis and some other things. So we really need to worry about exchange between the cytosol and the mitochondrial matrix. What has to happen? Well, it can't just diffuse. NADH, NAD+ can't cross over. Protons can't cross over. So they're not going to cross over by themselves. So we have to talk about transport systems that are going to allow us to move NADH, NAD+ around. So when we talk about this, right, you have to think about what tissues you're in and what you need. So there are really two shuttle systems that we talk about, and that's the shuttle systems in the liver and in the muscle. So they're going to be different because they have different roles. But the whole point is to get those electrons that were in the cytosolic NADH into somewhere in the electron transport chain. And so depending on which shuttle we use, we're going to link to a different part of the electron transport chain. Sometimes we'll go to complex 1, sometimes we'll go to Q. And if we go to different places in the electron transport chain, that means that the number of protons that are translocated is different depending on which shuttle system we use. So we're going to get different numbers of ATP per glucose molecule, depending on what tissues this is happening in. So we kind of have to keep that in the back of our minds. But NADH and NAD+ are not the only things that need to be transported. Remember, we just made ATP in the mitochondrial matrix. But where are we going to use ATP? Well, that's going to be in the cytoplasm. So we got a lot of things to do here. Number one, we got to export the ATP that we make. We need to import the ADP and the inorganic phosphate into the matrix. So there are really two ways that this happens. We have ADP and ATP translocase. For every one ATP that goes out, one ADP comes in. And we have a phosphate translocase. We actually have two different kinds. One's a symporter, one's an antiporter. The symporter brings an inorganic phosphate and a proton into the matrix at the same time, whereas the antiporter brings an inorganic phosphate in and a hydroxide ion out. And so I'm going to show you kind of how that works. So we'll talk about the ADP, ATP stuff first, and then we're going to get into our other shuttle systems because it's a little bit easier. So when we talk about this, right, we have to kind of understand the structure of ATP, ADP, and inorganic phosphate. We've kind of just like said the names, right? But when we talk about ATP, how many negative charges do we have on ATP? Well, we're going to, every one of those oxygens that does not have a hydrogen is going to have a -1. So we'll have a charge of -4 on an ATP molecule. ADP, we're going to lose one of the phosphates. So now we have 3. When we look at an inorganic phosphate is only one phosphate group, but only one of the oxygens is unbound to a hydrogen. So it only carries a -1. And so if you see either one of these written, PI, P with a subscript I, or a H2PO4-, it's the same thing. It's an inorganic phosphate. Okay. So we said there are two different ways that this can happen. We have an ADP and ATP translocase. So this, for every one ATP that comes in, an ADP, for every ATP that goes out, an ADP comes in, excuse me. So that would be our antiporter, right? Then you have a phosphate translocase. It translocates one inorganic phosphate and one proton into the matrix. This would be the symporter. There's also an antiporter, and I'll show you that, but the symporter is kind of more commonly taught. Okay, so when we look at translocases, we call this, it's kind of a funny name, we call this the rocking bananas. So if you think about this as being one banana and this as being another banana, I know it's kind of ridiculous, but it works. Okay, so this translocase is going to bind to ADP and ATP with different affinities. And the affinity depends on the conformation of these rocking bananas. And we know this because you can have different inhibitors that block different conformations, right? And so we know that there have to be different conformations. So if we look in the C state, right, where we're open to the intermembrane space. So here's our intermembrane space. We're open to the intermembrane space. ADP binds with very high affinity. So after ADP binds, it induces a conformational change. The conformational change now opens this protein up to the matrix side. So that's why they call it the M state. So the M state, once that conformational change occurs, it's no longer high affinity for ADP, so ADP gets released into the matrix. Now that M state, we're still in the M state, has a high affinity for ATP. But once ATP binds in the pocket, it induces a conformational change back to the C state. Now the C state doesn't have a high affinity for ATP, so it releases it. And then another ADP comes in, and so on and so on and so on. All right, so we can see that inducing conformational changes results in a difference in binding of ADP for ATP. So now I want to pause, and I want you to think about the charge differential. So ADP has a charge of -3. ATP has a charge of -4, right? So the electron transport chain is providing energy for this translocase by establishing a membrane potential that actually favors the export of a negatively charged, of a net negative charge, right? Because we're doing an ATP -4 for an ADP that's -3. So we're exporting a net negative charge in the direction of the more positive intermembrane space. So it's a really kind of cool mechanism. All right, so now let's talk about the inorganic phosphate. The inorganic phosphate translocase is much, much, much more similar to a channel. And that channel can be either a symporter or an antiporter. And this is not like our ATP ADP translocase. This one is electrically neutral. Okay, so how is it electrically neutral? Well, if I bring in an inorganic phosphate, and at the same time I bring in a proton, I'm bringing in a positive charge and a negative charge. They cancel each other out. So the charge hasn't changed in the intermembrane space or in the mitochondrial matrix, right? So that that's an electrically neutral translocation. The other way that this can happen is you can have an antiporter, to where we need to bring in our inorganic phosphate. That's really important. And at the same time, we're going to ship out a hydroxide ion. And so if a negative comes in and a negative goes out, it's an antiporter, but it's an electrically neutral antiporter. So it's all good. Okay, so now let's kind of put everything together, right? So when we talk about the number of protons that have to be translocated in order to drive our ATP synthase, right? We know we need 3 protons to move through our ATP synthase complex. They go from being in the inner membrane space into the mitochondrial matrix. And at the same time, we take an ADP and an inorganic phosphate and turn it into an ATP. So what did that cost us? Well, it also cost us a proton in order to be shipped in so that I can also bring in an inorganic phosphate. Most of the time, not all the time. Sometimes you can use that antiporter, right? So how many protons do you really need to synthesize your ATP? You really need 4 going from the inner membrane space to our matrix. So we get 3 of our protons through the ATPase. And then we have 1 proton that goes through our phosphate translocase. Right, so that's kind of a cool thing to think about. And then you also have to ship out your ATP, and at the same time you're shipping in your ADP, because those are the same. This is the rocking bananas model. Okay, so now we've talked about ATP and ADP. Now we have to talk about NADH. So remember that we said we have NADH that's made in the cytosol from multiple different places, but we've talked about glycolysis. So how are we going to move it? Well, it depends on what tissue you are in. If you are in liver cells, you are going to use the malate aspartate shuttle. This takes our electrons from NADH that's cytosolic, and it turns it into NADH that is mitochondrial. So if I have NADH that's mitochondrial, where is that NADH going to drop off electrons? It's going to drop off electrons to complex 1. So we know that our NADH is not going to cross that inner mitochondrial membrane. So we have to be able to somehow get NADH from the cytosol to the mitochondria without actually moving it. It's kind of a weird thing, right? Okay, so when we talk about this, why is this important? Yes, we're going to get some electrons in order to drive the electron transport chain, but we really made a lot in the citrate cycle. So do you really need them to drive it? Not so much. What's really important is that we regenerate our NAD+, because if we don't have any NAD+, is glycolysis going to happen? No. We do not want to inhibit glycolysis. So if we can regenerate our NAD+ in the cytosol, that's what we need to do. So we're going to take our NADH that's cytosolic, convert it into mitochondrial, then drop those electrons off at complex 1. This process, do you need to know this? Yes, you need to know this. This process is what gives you the 2.5 ATP per NADH ratio, right? And so this is NADH that are found in the mitochondria. So you got to be very, very careful about that, because we're gonna talk about if it's not in just a few minutes. Okay, so here's our process. We have glycolysis. Glycolysis has used NAD+, and it's made NADH. So we know that we have this enzyme called cytosolic malate dehydrogenase. Using this enzyme, we can take oxaloacetate, turn it into malate. Now, there's a transporter that will transport a malate molecule in at the same time that it transports an alpha ketoglutarate out. So that malate is what's carrying our electrons. This is why we call it the malate aspartate shuttle. It's what's being transported in and out of the mitochondria. So malate is going in. Now, we have a similar enzyme. We had cytosolic malate dehydrogenase. Now we have mitochondrial malate dehydrogenase, where we're going to take that malate and turn it back into oxaloacetate. Well, how are you going to do that? At the same time, you're going to take NAD plus and turn it into NADH. Now that we have that NADH, we can drop off those electrons to the electron transport system. And in order to make that happen, that regenerates our NAD plus. So we basically have these pools of NADH and NAD plus that are separate. One pool's in the mitochondrial matrix, one pool is in the inner membrane space / cytosol, because the outer mitochondrial membrane is very permeable. So it's very easy for things to pass, right? Okay, so now oxaloacetate is okay, but we got to get the substrate out so we can keep this cycle going. We want this to be a circular cycle. So in the third step, we're going to have a transamination of oxaloacetate by our mitochondrial aspartate aminotransferase. And so we're going to take oxaloacetate and glutamate, and we're going to turn that into aspartate and alpha ketoglutarate. Look at this. These are all substances that we already know are in the mitochondria, which is like fantastic. Makes it so easy. Okay, so that aspartate can actually be transported out of the mitochondria. So we had, at the same time that we're transporting our aspartate out of the mitochondria, we're also going to transport glutamate into the mitochondria. Well, why do we want glutamate in the mitochondria? Because we needed it for that reaction. The same thing is true about the alpha ketoglutarate. Where did we get this alpha ketoglutarate that we used as an antiporter? Well, where did it come from? We just made it here, right? So you can see how interconnected everything is. And so you have these pools of alpha ketoglutarate and glutamate. And the same thing is true on the cytosolic side or the inner membrane space side, however you want to think about that. Once I make aspartate, I can turn it back into oxaloacetate using alpha ketoglutarate and cytosolic aspartate aminotransferase. And I'm going to make glutamate. Well, that glutamate is going to be used in the transport mechanism here. The alpha ketoglutarate comes from this one. So it's a very, very nice circular pathway where everything is regenerated. So what drives this? Like, everything's so interconnected. What drives this? It's really driven by the NADH and the NAD plus ratios. So depending on what you have more of, what's going to happen. So if you need to generate more NAD plus, then you're going to start this process at that point. But if you have lots of NAD plus, you don't need to do this. So it's all about those ratios. Okay, now, what tissue type was this in? Go back and remember that this was in the liver. So go ahead and put yourself, this is the liver, right? The liver is going to be that malate and aspartate shuttle. But what happens in other tissues? So muscles are really sort of an interesting thing to think about, because if I want to, let's say, go for a run, and running now isn't like a survival thing, but it used to be for us. So if my muscles can't get enough ATP in order to work, then I'm going to get eaten by the bear that comes in the classroom, or whatever it is. So the process of shuttling NADH from the cytosol into the mitochondria for muscle tissue has to happen in a much, much, much quicker way. But if you want it to go fast, it's therefore going to be less efficient. So I'm not going to get as much ATP per molecule of NADH anymore. So whereas up here, I'm getting 2.5 ATP per NADH, if we come down in the muscle, I'm now only going to get 1.5 ATP per NADH. And I'm going to tell you why that is in just a second. So it's very similar, where we're going to deliver those electrons from the NADH that's in the cytosol to our mitochondrial matrix. But we're going to use a different intermediate, and that intermediate is FAD. So if you remember back to your electron transport chain, where do electrons go if they're residing within FADH2? They're going to go to coenzyme Q. So they're like entering in the middle of the electron transport chain. So there's no way that they can make the same amount of ATP as going through the whole electron transport chain. And so this happens really quickly because it relies on only 2 enzymes, and they're isoenzymes. Whoops. They're isoenzymes, which means they have the same function, but they're located in different cellular compartments. So what we're going to see is we have 2 different versions of, it's called glycerol 3-phosphate dehydrogenase. One cytosolic and one is mitochondrial. So we're going to go through glycolysis. We've got NAD+. We're going to use up that NAD+ in glycolysis. We're going to make NADH. We are in need of NAD+. So that NADH is going to be oxidized by our cytosolic glycerol 3-phosphate dehydrogenase. We're going to turn it into glycerol 3-phosphate. That glycerol 3-phosphate is going to be transported directly into that intramembrane space. Once it's in the intramembrane space, now we have this mitochondrial enzyme that will convert it into dihydroxyacetone phosphate, DHAP. We remember this from glycolysis. This is our glycolytic intermediate, right? This is reaction 5, where we took fructose 1,6-bisphosphate with our aldolase, and we made dihydroxyacetone phosphate. And this is so, like, go back and remember these. Go back and review your glycolysis, because things are so interconnected, right? Okay, so now we have our dihydroxyacetone phosphate, but in the process of that conversion from glycerol 3-phosphate to dihydroxyacetone phosphate, what's happening is that it's being oxidized, right? So those 2 electrons are transferred to FAD, turning FAD into FADH2. Well, where did we learn that FADH2 drops off its electrons? We know that happens in complex 2 because that complex 2 is a part of the citrate cycle. But what if we didn't use the enzyme from the citrate cycle? What if we used mitochondrial glycerol 3-phosphate dehydrogenase? Then it's going to drop it straight off to Q. Then Q will bring it to complex 3. So this is extremely fast, extremely fast, but we're only dropping it off at Q. So we're basically starting those electrons off at complex 3. That's the first complex, the first part of the pathway that can actually transport protons into that intermembrane space. So that's a kind of important thing to remember, that this is in the muscle. So it goes fast, but it's not as effective. So I want you to remember about Q. Q is very special because Q can pick up electrons from 3 different places. We know that Q can pick up electrons from complex 1, right? Where did those electrons come from that were in complex 1? Well, they came from NADH that's located in the mitochondria. Doesn't matter how that NADH got there, but if it's in the form of NADH and it's in the mitochondria, those electrons are going to be dropped off when NADH is oxidized, and they're going to be dropped off to complex 1. Q can also get electrons from our glycerol 3-phosphate dehydrogenase. And we know that this happens in muscle tissue, right? Whereas in complex 1 or NADH, this could come from, let's say, the citrate cycle. CITRAT, citrate cycle. It could also come from our transport system in the liver, right? So either one, it's dropping it off at complex 1. Whereas if we're in the muscle, we want to go quicker. So we're going to transfer that NADH, that cytosolic, into FADH2 in the mitochondria. And that results in dropping off at Q. And the third one that we haven't talked about yet, but we will when we get to fatty acids, is the ETFQ oxidoreductase. And so that's just all about fatty acids. So you just kind of have to like know that one, and then we're going to talk about him later. But you have to know he's there and he works, because that's going to be really important later. Okay. So let's kind of look at a breakdown here. This is really nice. And we're going to review this again in class when we have some time to kind of like work through it together. But so if you first look at just this step here, this is glycolysis. This is in the cytosol. And in glycolysis, what do we generate? Well, we use up, regardless of whether we're in the liver or whether we're in the muscle, doesn't matter. We're going to consume 2 ATP and we're going to produce 4 ATP. So regardless of whether we're in the liver or in the muscle, we're going to get a net of 2 ATP from glycolysis. But what else do we get? Well, we get 2 NADHs. Where do those 2 NADHs go? Well, we just learned that it depends. It depends. If I'm in the liver, then those 2 NADHs are going to go through the malate aspartate shuttle, which means I'm going to take 2 NADH times 2.5, and I'm going to get 5 ATP. Now, if on the other hand, I'm in muscle cells, I'm still going to make the 2 NADH, but I'm going to go through a different shuttle system, which is the glycerol 3-phosphate shuttle system. So those electrons there are going to enter into the electron transport chain later on, and I'm only going to make 1.5 ATP per molecule of NADH. So I only make 3 ATPs for my 2 NADHs. So you see how this is an either or. The green is what happens in the liver, the blue is what happens in the muscle. Now, you also have to remember that we have pyruvate, and here's our pyruvate. So it's our pyruvate dehydrogenase and the citrate cycle combined together, because they're both happening in the mitochondria. And if we add up all of the NADH that we have produced, we're going to get 8 molecules of NADH. We're also going to make 2 molecules of FADH2, right? Those 2 FADH2 are going to be multiplied by 1.5, whereas the 8 NADH are multiplied by 2.5. So regardless, right, you're still going to make the same amount. From the mitochondrial NADH, you're going to make 20 ATPs, and from the mitochondrial FADH2s, you're going to make 3 ATPs. And then you also have to remember that in glycolysis, you're going to make GTP as well. So, and you're going to go through the citrate cycle twice, right? Because this is starting with 1 molecule of glucose. So to metabolize 1 molecule or oxidize 1 molecule of glucose, you're going to have to go through citrate twice. So you're going to get those 2 GTPs, and 2 GTPs are equal to 2 ATPs. So from glycolysis, we got a net of 2 ATP. From citrate, we got GTP straight to 2 ATP. Then from pyruvate dehydrogenase and citrate cycle, we're going to get 23, which is the 20 plus 3. So if I add all of those up, 23 plus 2 plus 2, doesn't matter if I'm in muscle or if I'm in liver cells, I'm still going to make 27 ATP. Now, what's going to be different is my NADHs, that were generated in glycolysis? Are we in the liver and we're going through the malate aspartate shuttle, or are we in the muscle and going through the glycerol 3-phosphate shuttle? If we are in the liver, we're going to go through the malate aspartate and we're going to get 5. So we're going to do 27 plus 5, and we're going to get a net of 32. If, on the other hand, we're in muscle and we're way more worried about making ATP quickly than getting the maximum amount of ATP that we could get, then we're going to make 27 plus 3, and we're going to get 30. So this is why sometimes in books you're going to see different yields of ATP. So this is one of those things you should be able to justify the number of ATP, where they all came from, all that kind of stuff. And we'll talk about it in class some more. Okay, 11.5, we're almost done here. This is regulation of oxidative phosphorylation. So we've already talked in great detail about regulation of glycolysis, of citrate, and so much of it depends on the energy charge of the cell, right? So ATP, ADP ratios, and the ratios of NADH and NAD+. And that's true also in oxidative phosphorylation, right? So the ratio of NADH is really going to control the flow through the electron transport chain, because if you regulate the citrate cycle, you regulate the electron transport chain. If you regulate glycolysis, you're going to therefore regulate the citrate cycle and the electron transport chain, and therefore oxidative phosphorylation. So if you kind of look, I know this is pretty small, but a lot of these things you already know. I'm not introducing too much new. If you have a lot of inorganic phosphate, that's telling me that my energy charge is really low. I don't have a whole lot of ATP. So any glucose that comes into the cell, I got to phosphorylate it and trap it. Then we know that phosphofructokinase 1 is a key regulatory step in glycolysis, regulated by energy charge. Our pyruvate kinase, our pyruvate dehydrogenase reactions, those are both very highly regulated, right? So pyruvate kinase is our last step in glycolysis, whereas pyruvate dehydrogenase is that link between glycolysis and the citrate cycle. And then that first step in the citrate cycle of citrate synthase, highly regulated by both energy charge and NADH. And then we have our isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase reactions within the citrate cycle that are highly regulated, right? So the electron transport system and oxidative phosphorylation, it's all dependent upon everything that comes before it. So you have the ratios of all your different electron carriers and your energy charge of the cell that's going to regulate your oxidative phosphorylation. Okay. Oh, this is really cool. I love how your book includes a little bit of experiments in. So when we talk about the electron transport chain, we've talked about how you can bypass that electron transport chain. Remember how we talked about if you wanted to create heat, that you could create an uncoupler that would allow those protons to flow from the intermembrane space into the mitochondria without having to go through the ATP synthase complex. That was really in the beginning of the chapter. So the question is kind of, what's happening? How does this work? So if we take mitochondria and we give them succinate and we give them ADP and inorganic phosphate, and what we look at are two things. We're going to look at oxygen consumption. That is our blue line. So our blue line is oxygen consumption. And then our red line or pink line, whatever, is ATP synthesis. So if I give my mitochondria the things I need, okay, both oxygen consumption and ATP synthase start going up. Great. Now, what happens if I put in an inhibitor to the electron transport chain like cyanide? So if right here I put in cyanide, that's going to shut down the electron transport chain. Energy consumption shuts down, ATP synthesis shuts down. If I add an uncoupler, nothing's going to happen, right? There's no effect because the electron transport chain is stopped too early. But what happens, let's look at like part B, right? We're going to do exactly the same experiment. But in this case, instead of inhibiting the electron transport chain, now I'm going to inhibit the ATP synthase complex. So the electron transport chain is going to work. It's just the ATP synthase complex that's not going to work. And I'm going to do a very similar experiment. I give my mitochondria what they need. So I see an increase in O2 consumption. I see an increase in ATP synthesis. As soon as I introduce oligomyosin, excuse me, I introduce oligomyosin, ATP synthase stops. So that's where I see my leveling off of ATP synthesis. And I also see a leveling off of O2 consumption. But now, if I've inhibited the end of the pathway, if I add this 2,4-dinitrophenol, so this is my uncoupler again. If I add this uncoupler, then I'm basically bypassing the ATP synthase complex. So I'm still going to not make any more ATP, but my oxygen consumption is going to resume again because those protons can now flow. If you remember, they can now flow back across the membrane. This was my ATP synthase, right? And this was my electron transport chain. The protons moving up in here, and usually they have to go through my ATP synthase complex. But if I add my oligomyosin, ATP synthase complex won't work. So the protons would have to go through this uncoupler of 2,4-dinitrophenol in order to allow the mitochondria to keep running, basically. So this 2,4-dinitrophenol is kind of interesting. It was originally came on the market and people were like, oh, this is a great diet drug. And they thought it was a great diet drug because it can activate fatty acid oxidation. But what they didn't know is that it's also poisonous and it can kill you because it accumulates in cell membranes. And when it accumulates in cell membranes, it can attach to protons. So our oxygen here can pick up a proton. Once it picks up that proton, it can go back and forth, and it actually diffuses a little bit better once it's picked up that proton into the mitochondrial matrix and then drop off the proton there. So it acts as an uncoupler. It's not really a pore. It's not really a transport complex that's embedded in the membrane. It actually just picks it up and then diffuses. But it creates that short circuit that allows the protons back into the matrix. And yeah, not so good. Not so good if you want to survive. Okay. So when is it good to have an uncoupler? So I'll give you an example, right? If I bypass ATP synthase complex and I allow the protons to go through some kind of an uncoupler, and this is an example, this is called UCP1. UCP1 is also called thermogenin, and it controls thermogenesis. It's expressed very, very highly in certain fat cells. They're called brown fat. And they're called that because they have a higher level of mitochondria, and the cytochromes in the mitochondria give it a brown color. And so this is actually a good thing to have, because if I don't go through ATP synthesis, I mean, some of it could, you're not actually inhibiting ATP synthesis. You're just adding this uncoupler. So some of the protons will go and make ATP when needed. But if you have excess, then it's going to go through this UCP1 molecule. Okay, so why? Why would this be a good thing? Well, I want you to kind of look at tissue samples before you answer that. So if you look at normal adipose tissue, so as an adult, I guess I should start. Let me start it earlier. When you're born, about 5% of your total body fat is brown adipose tissue instead of white adipose tissue. And as you get older, you lose that brown adipose tissue. In humans. In other organisms like hibernating organisms, mice, squirrels, bears, you see that brown fat is retained, and it's actually um, in certain areas of the body like the head and neck. And I'll tell you why that's really, really important. But let's look at the structure real quick. So if you look at white adipose tissue, this is one cell, right? This is one cell. And that one cell is filled with one giant fat droplet. And the nuclei are like smashed along the sides. So that's why you see this kind of like darker area, right? The nuclei and all the other organelles are like smashed along the sides of the cells. Whereas if you look at brown adipose tissue has lots and lots and lots of small fat droplets. So each of these tiny, tiny circles that you see, those are droplets. But there's way more space for you to have lots of mitochondria. So you can tell that there's a color difference in the tissue, and that's why they call it brown, right? Okay, so why is this important in things like hibernating animals, right? At the end of hibernation, when they're supposed to wake up, norepinephrine is going to send signals to increase the expression of UCP1. So UCP1, the more we have of it, or the more the squirrels and the bears and the mice have of it, it's going to have more heat. So it's going to warm the blood that goes from the neck up into the brain, right? And so that's the end of hibernation. That's what stops them from hibernating. And that's what actually keeps babies a little bit warmer without having to shiver. It's a non-shivering way to retain body heat. It's kind of cool. Okay, last little cool thing here is inherited mitochondrial diseases. So when we talk about the mitochondria, we know that when the egg and the sperm cell come together, the mitochondria in the sperm cell are located in the tail. That's why they can like swim around. So when the sperm fertilizes the egg, the tail of the sperm drops off, and only basically the head is retained. And so all of the mitochondria and the growing embryo come from the mother. So all of the mitochondrial DNA is passed from mother to offspring because it's only derived from the egg. It's really kind of cool. So males and females can be affected by mitochondrial mutations, but only the mother can pass on the mutation, which is cool. So if you have mutations in any of the electron transport chain genes, which are located in the mitochondrial genome, right? There's a mitochondrial genome as well as a nuclear genome. Then you're going to have a problem with ATP production, right? Even if it's just a decrease in expression. And you don't have to memorize all this. I just want you to understand that mitochondrial diseases are inherited in a different pattern than most diseases. So, and the other thing really is to think about is that you're not just going to have one mitochondria. You know, we have one nucleus in every cell, but we don't have one mitochondria in every cell. We have 10 to 1,000 mitochondria. And so the severity of the disease is going to depend on the ratio of normal to mutant mitochondria. And how do you know which mitochondria are going to be affected or not, right? When the egg cells are made, there's random segregation of mitochondria into that developing egg and therefore that oocyte, right? So it's very, very, very variable compared to nuclear diseases. And so most of the time you'll see effects in neuronal cells and skeletal muscle cells because those are the two cell types that consume so much energy. So I hope that was really interesting. And that ends our chapter 11, but we got some activities to do in class. All right. See you then.